Hello, everyone, and welcome to today's webinar titled New Challenges for SARS-CoV-2 Diagnostics, Viral Load Testing and Variant Strain Genotyping. This webinar is part of the Coronavirus Virtual Event Series. And I'm Susie Valdez of Laboratories, and I will be your moderator for today's event. Today's educational web seminar is presented by Laboratories and sponsored by LGC SeraCare. For more information about LGC SeraCare, go to seracare.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive and we encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Q&A box and click send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on that Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free education credits. Click on the Continuing Education Credits tab located at the top right-hand corner of your presentation window and follow the process to obtain those credits. I now want to introduce our presenters today, Dr. Russell Garlick, Chief Scientific Officer at LGC Clinical Diagnostics, and Dr. Nathan Ledeboer, Professor and Vice Chair of Pathology and Medical Director at Medical College at Wisconsin. For a complete biography of our speakers, please visit the Biography tab at the right of your screen. Gentlemen, welcome to both of you. You may now begin your presentations.
Well, thank you very much for the kind introduction today, and thank you for joining us on this webinar, exploring testing options and exploring some of the limitations and benefits of testing in the SARS-CoV-2 pandemic. This webinar will focus on viral load testing and the potential utility of viral load testing, as well as identification of variant strains and genotyping. My name is Nate Ledeboer. I'm a professor in the Department of Pathology at the Medical College of Wisconsin, and I also serve as medical director of the microbiology and molecular diagnostic sections for Wisconsin Diagnostic Laboratories and Froedtert. As we start to think about the SARS-CoV-2 pandemic, and as we start to think about the potential role of viral load testing, there are a number of important considerations that we need to think about and that we need to consider. And one of the biggest challenges that laboratories have encountered throughout the pandemic is getting access to testing, and then really addressing the question of what is the best test for detection of the SARS-CoV-2 virus. Well, we're not going to tackle that question today. It's important to recognize that there are a number of different assays that are available for the detection of SARS-CoV-2. The mainstay of testing has largely been PCR, or nucleic acid amplification testing. It's the gold standard. It's the most sensitive method. It offers a great deal of variation on the specimen types that we can work with, including nasopharyngeal specimens, oropharyngeal specimens, nasal swabs, saliva, and even stool. But the challenge with PCR, in most cases, is that it's lab-based. We need lab experts to run the test, and there have been very few options for point-of-care testing or testing for laboratories that don't have big, large instruments to handle high volumes. It's also important to recognize that while PCR is the most sensitive methodology, it tends to also be the most expensive. A number of other methods have been developed. These include a resurgence of antigen testing. While antigen testing has been reported to have variable levels of sensitivity, for symptomatic patients, most studies have now concluded that antigen testing is between 80 and 85% sensitive compared to PCR. Antigen testing has the benefit of being very amenable to the point of care and can be either read visually or via an instrument. We've also seen the application of antibody testing to the detection of the SARS-CoV-2 virus. And this has proven to be very useful in assessing the rates of infection in individual populations, in understanding disease prevalence in a population, and more recently, in assessing vaccine exposure in individual patients. So while these are all important diagnostic modalities for the detection of SARS-CoV-2, for the purposes of our discussion today, we're largely going to focus on the application of PCR. And if we look to the IDSA for their recommendations on where we should use PCR, they recommend the use of PCR or NAT testing in symptomatic individuals suspected of having COVID-19. They also recommend its application to a number of asymptomatic individuals, including those with known or suspected infection, including those patients where the results of the test will impact isolation, quarantine, or personal protective equipment, as well as asymptomatic individuals where results can dictate your eligibility for a surgical procedure or can inform administration of an immunosuppressive therapy. So even the IDSA recognizes just how important PCR is for the detection of the SARS-CoV-2 virus. Now, one of the things that many laboratories have encountered throughout the pandemic is the need to be able to provide very, very large volumes of testing. Our laboratory, for example, went from performing about 100 respiratory tests per day prior to the pandemic to, at its peak, performing more than 8,000 tests per day. This put a huge strain on the supply of available testing reagents, and it meant that laboratories needed to implement multiple methods in order to address the needed volume. This meant in many laboratories that they weren't doing one PCR or two PCR methods, but they may have been doing as many as 8 different PCR methodologies. And when we come to doing 5, 6, 7, 8 PCR methods, we have to recognize that there is a great deal of variation across individual tests based upon the manufacturer and the limit of detection of those tests. And this was nicely summarized on the FDA website in an evaluation of the limits of detection of available EUA-approved assays. So in this study, the FDA's Center for Diagnostics and Radiation Health sent out a SARS-CoV reference panel to manufacturers with an EUA-cleared test. The panel had 4 unknowns and 1 heat-inactivated strain of the SARS-CoV-2 virus, which was diluted to a reference standard of 1.8 times 10 to the 8th units per mil. The FDA then assessed the limits of detection across each manufacturer's tests based upon this panel. And they found significant levels of variation across each manufacturer, with the limits of detection ranging from 180 to 600,000 new data units or NDUs per mil. There were approximately 10 tests with a limit of detection below 1,000 NDUs per mil. An additional 26 tests with an LOD between 5,000 and 10,000 NDUs per mil, and there were more than 10 tests with a limit of detection of 180,000 NDUs per mil or greater. Now, while the individual numbers don't matter, it is important to recognize that the variation in LOD across each of these manufacturers' tests ranged between 1,000 and 180,000, or more than 2 logs. And that's important as we start to think about asking questions around quantitation. It's important we understand our tests. It's important we understand how low they go, and it's important to understand how reproducible they are. So why do we care about quantitation? Why is it of interest? And what is quantitation? We all know that we've been doing quantitation of nucleic acid in patient specimens for decades. It's a mainstay of managing HIV patients. We routinely use it in the management of cancer patients. We routinely use it in managing immunosuppression of patients and looking at viral loads. But it really begins with the CT value or the cycle threshold value. And for the purposes of our discussion today, the CT value is defined as the point in which our real-time PCR curve becomes logarithmic, or as you can see on the top graphic, where the line becomes straight. If we use a standard curve of known quantitative standards, we can begin to assess the amount of virus that may be present in a patient specimen. So to establish our standard curve, we take known standards, beginning with a low quantity of virus and ending with a high quantity of virus, and we run individual PCRs on those standards. You can see in this top graphic, the lowest standard tends to be on the right-hand side, has a CT value of 30, and the standard that contains the largest amount of virus has a CT value of approximately 12. We then plot those CT values on, and as you can see that on the graph on the bottom, against the known quantity of virus. And you can see we have a very nice linear line. We then go back to 8th grade algebra, and we use the old equation of y equals mx plus b to determine the viral load by solving for x. Y equals the CT value. We know the CT value from our PCR test. We input that CT value into our equation. M and B are standard from our best fit line, and we solve for x. And in solving for x, it helps us to determine the viral load of that individual patient. Again, these are things we've been doing for decades. When we manage immunosuppression, when we manage transplant-associated infections, when we manage HIV. We're now beginning to ask the question, is this of significance as we apply it to the SARS-CoV-2 pandemic? And some studies would say, absolutely, it's of significance. And this is a study published by Magleby and colleagues in Clinical Infectious Diseases. And in this study, the authors suggest that patients with a higher viral load or lower CT value were more often at risk of death, as opposed to patients with a lower or with a higher CT or lower viral load. So as you can see from this graphic, patients with low CTs indicating high viral loads had upwards of a 50% chance of dying, versus patients with a lower CT or with, a higher CT or lower viral load had a lower chance of dying. So it's studies like this that really push us to ask the question, can we use real-time PCR data in order to report viral loads, and is that of significance? Well, there are a number of important considerations that we need to think about when potentially reporting quantitative values for respiratory viruses. And this really begins at the time of specimen collection. The easy thing of doing viral load testing, for example, in blood, is that blood is fairly homogeneous. The presence of the virus, no matter what portion you sample, what vein you sample, is likely to be very similar. When we think about respiratory viruses, variation in collection technique can significantly alter the amount of virus that you detect. For example, if we collect a nasal swab as opposed to an NP swab, we may get different levels of virus in the nasal swab versus the NP swab. The amount that I scratch against the nose or the amount of pressure that I apply to the swab may result in different quantities of virus that may be released. We've gone to and utilized different suppliers for transport media. We've looked at using saline for transport of specimens. Transport media can have a significant impact on PCR efficiency and the amount of virus that we detect. The amount of transport media that's included in our vial can have an impact on viral load. The amount of time that it takes to get to the laboratory, and if that specimen was maintained properly, can have a big impact on viral load. What I'm getting at is that in many cases, the collection and detection of respiratory viruses has largely been designed around the concept that these are qualitative assays. They're designed for reliable detection of an individual pathogen, and they're not necessarily designed for reliable quantitation. When we think about moving from qualitative to quantitative, we really need to start to think about including reference standards. This ensures reproducibility. It helps us to ensure precision of our assay. It also helps us to look at variation in viral load or variation in CT between methods, between manufacturers' tests, and across multiple lots of tests. It helps us to ensure that our targets quantitate similarly, because we know that there can be different amounts of E gene as compared to nucleocapsid, for example. And we also know that extraction platform can account for significant levels of variability. And this was all nicely highlighted in a study published by Dan Rhodes and colleagues as a part of a statement from the College of American Pathologists Microbiology Resource Committee. And in this particular study, they looked at CT values for gene targets and manufacturers across the same batch of testing material. So we all know how CAP proficiency testing works. We get unknown samples, we test it like we do a clinical sample, and we report results back to the CAP. We report that in addition to the method that we report. And you can see that at the time that this data was collected, the largest platforms included the BD MAX platform, the CDC test, the Gene Expert, the 6800, as well as a host of other different instruments. And each of these tests utilized different genes. And while we saw a relative scattering of CT values all around the 30 range, we saw significant variation across manufacturers' tests. So for example, with the CDC assay, across 67 laboratories that were performing the CDC method for detection of the SARS-CoV-2 test, some reported CT values as low as 28, while others reported CT values as high as 41. For the Thermo Fisher method, the average CT was approximately 31, with some laboratories reporting CT values as high as 35, and others reporting them as low as 25. A variation of 10 CTs is quite significant and can represent more than a log difference in viral load. So understanding how reproducible our tests are, understanding what our tests quantitate, and how our tests quantitate are really crucial to begin to assess whether there's any utility to reporting viral loads. We also need to think about the frequency and the need for controls, because it changes significantly based upon a quantitative test as opposed to a qualitative test. For quantitative tests, we need to make sure that we verify the limit of detection and really know, just like the lady here doing the limbo underneath her car, we need to know how low our test goes. We need to understand how frequently we run controls. At minimum, we need to do a positive and a negative control for each run. For quantitative tests, we also need to be running calibrators to ensure that we get reproducible quantitation. So we partnered together with 6 other medical centers to begin to look at, can we accurately quantify virus, and can we develop controls to be able to help in assisting quantitation of the virus. So this really began to ask the question, can we move from semi-quantitative RT-PCR by quote unquote reporting the CT values to can we generate a standard curve? Can we report in copies per mil? And is that reproducible across multiple laboratories and across multiple platforms? To do this, we developed a series of calibrators which ranged from 300 to 1 million copies per mil. And we assessed the linearity of a panel using both real-time PCR and digital PCR. And we did this across 7 labs with geographical diversity in the United States. So the first thing that you need to do when we develop a panel of calibrators is really to determine, can we build a number of different targets and accurately measure those targets using both digital PCR and real-time PCR. And you can see from this graphic, the panel that we built began at 300 copies per mil and ended at 1 million copies per mil. And what we measured was actually quite similar, where we had a target of 316 on the lower end, we detected 393. Where we had a target of 1 million on the upper end, we detected 1.1 million. So we had a pretty linear panel that we were able to develop. So once we've established an appropriate reference panel that is linear across the range that we wanted to test, the next question really comes down to, is this panel reproducible across multiple laboratories? And is it reproducible across multiple platforms? So we recruited 7 sites running multiple different test methods in order to assess this question. These sites included laboratories running the Abbott Alinity, the Abbott M2000, the CDC method, the Roche 6800 test, the Cepheid Gene Expert, the QIAstat, as well as a site running an LDT using Sarbeco reagents. We then asked each site to perform the panel, repeat the panel, or to run the panel, and report CT values back to the central collection site. What we found was that across individual platforms, the panel was very linear. So the panel did what it's supposed to do, and it found high viral specimens and adequately distinguished those from low viral load specimens. We also found that when run on a single platform, the results were quite reproducible. So look, for example, to the lower set of lines on the graph. You'll see 3 individual sites ran the M2000 assay. And regardless of the site performing the test, the CT value that was reported for each known standard was very, very similar. Now, quite interestingly, we also did notice that the M2000 ended up reporting CT values that were a bit lower than the other platforms. And this is largely because it runs its first 10 cycles of PCR in a silent mode or in a blacked out mode where the labs don't see the results, and it only begins to report CT values after that 10 cycles of report. So if we correct for that on the Abbott M2000 platform and we normalize across all of our individual sites, you'll see that the overall CT values that were reported across our sites were reasonably reproducible, and they generally fell within a range of about 5 CTs. If we then translate that into viral load detection, the data becomes even more interesting. So for the viral load detection, we had 4 laboratories run the tests. The calibrators were run across 2 digital PCR platforms, as well as on the M2000 and the Sarbeco qPCR assay. And again, if we look at viral load detection, you actually find that regardless of the method that the test is run on, the individual laboratories report viral loads that are very, very similar to each other and are within the expected level of variation across the laboratories. So we certainly were able to, at least early on, demonstrate that when we include the proper controls, when we include the proper calibrators, and if we can standardize for all of those other things that we talked about, viral load testing is a possibility should the field continue to move towards needing viral loads reported on individual patient results. I'll thank you for your attention today, and I'll look forward to hearing Russell's portion of the talk. So I'll hand things now back to Russell.
Thank you, Nate, for that terrific presentation. Now I will speak on the emergence of SARS-CoV-2 mutations in genotyping. I'll speak about the mutations in the background, talk about assays, and of course, reference materials for assay development, validation, and QC. And then both Nate and I will be available for a Q&A session. So SARS-CoV-2 mutations. Let me first start with a discussion on nomenclature. It can be very confusing, so I think we should be all on the same page as far as how we're using the terms mutation, variants, and strains. A mutation it refers to an actual amino acid change in the sequence. Case example is D614G, which is a switch of aspartic acid to a glycine substitution at position 614 of the spike glycoprotein. A variant is a change in the genomic sequence. And in the case of D614G, it's actually a nucleotide change at the genomic position 23403, again in spike protein. The strain is a variant or a collection of variants that demonstrate a different phenotype. Could be antigenicity, transmissibility, infection, virulence, or escape from various therapeutic agents, especially monoclonal antibodies. Examples are B117 in 501v2 and B1128. And we'll get into more details of this as we go through the presentation. All of these different strains include one of the mutations, D614G, plus many other mutations. And very importantly for today's discussion, how it impacts laboratories, what does the CDC say about these classification of variants? Variants can be of interest, VOIs. These where mutations can span multiple genes and regions. They're being watched for concern and potentially high consequence. So a variant of concern is a variant that could induce a change in antigenicity, transmissibility, variants. Any of these could potentially become a variant of high consequence. So the VOCs are being very watched closely. And all of the ones we're following today are actually in the S gene. Variants of high consequence can lead to a failure of diagnostics, that is, the assay doesn't detect them, can cause a significant reduction in vaccine effectiveness, it could be causing high breakthrough in vaccine administrations, or could have low vaccine-induced protection against severe disease, significantly reduce susceptibility to multiple emergency use authorized or approved therapeutics. Can cause more severe disease or increased hospitalization. So these are really key consequences of these mutations in the SARS-CoV-2 virus. Fortunately, today, none have been classified as VOHC, or variants of high consequence. I like this website because this gives you a really well-researched and referenced report on each of these variants. This is the covariants.org website. And as an example, I'm showing you the variant E484. And as you go to this website, you will see all of the recent publications of importance that talk about changes in vitro characterization of this particular strain. And it can cause, in this first one here, this mutation may make significantly reduced convalescent serum neutralization with a nice reference. So I'd really highlight this website as a place to go to learn more about a specific variant. Now, where are we today in the United States? This is rapidly changing. This is from the CDC website, and it shows the percent of the total viruses identified by NGS sequencing across the United States. And as you can see, between January of this year, 2021, and mid-March of this year, the original Wuhan strain of B.12, you can see is actually declining in prevalence within the US population. And other variants are actually increasing. The B117, which was originally isolated in the UK, as you can see, it's dramatically increasing over time. If you look at the current data in my home state of Massachusetts, B117 is about 50% of the cases right now. Now, this table shows the different variants classified in the percent identified across the US. It also shows whether it's a variant of concern or a variant of interest. Here's the B12, the original Wuhan. These are the two California variants, B1427 and P1429, as well as you can see down, P1 is actually growing severe, another variant of concern down here at the lowercase. So this particular CDC website shows the cumulative across the US. I would highly recommend for any of the laboratories, keep a close eye on your regional variants that are circulating in your area as well. So some more background information on SARS-CoV-2 mutations. Viruses are mutating all the time. We know that, and it's been going on for over a year right now, and will continue to go. Some of these variants become dominant due to selective advantage of the virus, whether that's binding to the ACE2 receptor. There are multiple approaches to assess these mutations, whether they're VOIs, VOCs, or VOHCs. For the most part, they're in vitro detection methods right now. The US has created this SARS interagency group called SIG, which is now integrating data and activities between these major organizations within the US to focus on these variants of concern and really dive into data sharing and make sure we're starting to get ahead of the impact of these variants on therapies and diagnostics. The FDA has reported that for in silico analysis, if it predicts a performance change of greater than 5%, then the laboratory should use that assay and test with clinical samples or use synthetic RNA that includes these representative mutations to determine how it impacts the assay. And notification is required to the FDA if there's a significant performance change. For example, a change of 3x increase in the limits of detection. Also on B1315351, it shows a 6 times lower neutralization than the Wuhan strain. And this is important because this data that you'll be seeing, this is only neutralization titer, but it doesn't really reflect the immunological status of the patient. It doesn't reflect what the memory B cells or the T cell activities are invoking for immunity. But E484K is considered an escape mutation for therapeutic antibodies. I'll say a little bit about that in a minute. So let's be very aware from a laboratory perspective. We should be looking very carefully at our assays and be testing our assays with synthetic RNA, or I will show you in the subsequent slides here how you can use the Accuplex technology to test if your assay can detect these various variants. This chart shows the structure of the binding of the SARS-CoV-2 spike receptor binding domain protein directly to the ACE2 receptor. The virus uses the ACE2 receptor to gain entry into the cell for reproduction. And we have so much knowledge now about the exact binding of the virus to the structural proteins. And as more information is being generated, in this case, it's a bioinformatics approach, looking at the binding of the spike protein to the ACE2 receptor and the potential changes that you can have with these mutations. So here's the Wuhan strain of the SARS-CoV-2 binding to the ACE receptor, and here's the E484 site, that amino acid site. And over here in this particular part of the section shows where the E484 is mutated to a lysine group, and that impacts, it actually makes the binding a little bit stronger, showing more infectivity of this particular strain to the ACE2 receptor and causing greater infection in the population. So we know a lot about the particular mutations that impact not only on the assays, potentially on the assays, but also on the therapeutics that are being in development. The FDA is also providing alerts for variants as they impact monoclonal therapy. This is a very successful drug for Lilly. It's called bamlanivimab, and it was introduced as a monotherapy for COVID-19 patients who are mild to moderate, but potentially with multiple cofactors. And The data shown in this alert is pseudovirus neutralization data for SARS-CoV-2 and the variants and the substitutions. So on this column here are the different lineages from the 117, South African, Brazil, etc., California, and the key substitutions that were tested. And as you can see, no change with the UK 117, but significant change in the pseudovirus neutralization data for these various strains. And consequently, the FDA has provided alert not to use this drug as a monotherapy, but fortunately, it can be used very successfully with a combination of other monoclonal therapies, especially the bamlanivimab with other Lilly monoclonal antibodies. So a very proactive approach by the FDA providing these alerts. And what's important for us laboratorians is to make sure that we're very much aware of what the FDA is saying as far as neutralization to other monoclonals that could be on the marketplace. So not only we have to pay attention to what the FDA is saying regarding the laboratory tests, but also the impacts on therapeutics and potentially on vaccines as well. Now, considerations for the laboratory. This is an excellent publication from Filkins, Sorrell, Scogins, and Jason Park, University of Texas, published in Clinical Chemistry. And it's targeted to laboratories and manufacturers about revalidating their assays to confirm limits of detection with the emergence of virus variants in our regional areas. Now, quote from the paper, genetic changes may not cause a total loss in signal, but may lead to a decrease or increased detection efficiency. Therefore, laboratories should consider confirming assay limit of detection when the new variant becomes prevalent within the population tested, or quantitative reference materials become available to determine the analytical sensitivity. So laboratories have to be very proactive, whether it's an IVD assay or an LDT assay. And the authors point out that IVD manufacturers actually do not test for mutations with real samples or standards, but really rely on in silico analysis. And it's up to the laboratory to take the lead and do these testings. And case in point, the B117 impacted both the TACPath thermoassay and the Linnea Applied DNA Sciences PCR assays, and there were S dropouts. Since these assays look at three different regions, just having an S dropout didn't impact the positive or negative call for these particular tests, but it was the original identification of how these variants were impacting the assay. So a very good reference to look up is this Clinical Chemistry by the Park Laboratory. Now let's move on to assays. So with SARS-CoV-2 genotyping, let me just provide an update. Today, there are no emergency use authorized genotyping assays using either RT-PCR or sequencing. There are assays from these particular companies that have UAs using NGS, but they're qualitative assays. They're not screening, they're not genotyping. It's a yes, no. Is a person infected or not? NGS has a number of approaches used for screening to identify these variants. For example, Thermo just launched a 22 variant panel, which is RUO label, uses RT-qPCR on the TACPath assay, and runs on an ABI PCR instrument and uses the QuantStudio readout. So here's one of the first RUO assays that you can actually adapt to identify these variants. Again, it's for genotyping. And there are many laboratory developed tests using RT-qPCR. They're targeting some of these VOCs and emerging VOHCs as well. Predominantly, de novo sequencing is used today either on the Illumina platform with the MiSeq instrument or the NextSeq using the Arctic protocol, or the Oxford Nanopore using the LandPore high throughput COVID-19 assay. So a couple of different ways to look at really getting a deep view into the variants within your regions. This is a snapshot from BioSearch, LGC BioSearch, one of my colleagues' companies, and This shows the specifications and comparison of probe-based RT-PCR versus whole genome sequencing. I'll just point out a couple points here. So you can go to this website and get lots of information about the primer probe materials that are available from BioSearch. But the variants detected in the probe is just, you have to design the assays for what you're looking for. With whole genome sequencing, you see them all. Design requirements, every variant has a new design. This is none required since it's whole genome sequencing. This is fast, sequencing slower. This is a standard, well accepted methodology, greater expertise required here, a little bit higher cost on sequencing. Right now it'd be high throughput on the genotyping assays and lower throughput on sequencing assays. But as we advance variant testing, I think you'll see more sequencing-based approaches get to high throughput and at lower costs as we continue to see the importance for detecting variants in the population. Now let me move to reference materials and the role here for LGC SeraCare. We've been using the Accuplex technology since the beginning of the pandemic to produce recombinant SARS-CoV-2 virus that can be used for assay development, assay validation, and QC. And very briefly, what we do is we take the sequence of interest, whether it's the original Wuhan or these variants that we've been talking about, we clone them, and the RNA is introduced into a mammalian cell culture for production. This is industrialized at scale, so we can make lots of materials for many different laboratories and companies. The Symbis virus that is used to produce the sequence is actually produced in the cells and is a fully encapsulated virus, so it has to go through a full extraction. And these virus particles are replication deficient. But we also go through a heat treatment step to ensure, kind of a double safety approach that they're non-infectious. And what we've been able to do is now we have the full genome SARS-CoV-2 with the Wuhan lineage. And what we've done now is we've added new sequences that are applied to these variants of interest. Let me show you that next. So for example, this is the region within the S gene, the spike protein. This particular strain, we've actually included all of these different variants in the mix, so that there's 2 deletions and a number of these mutations here shown by the letter changes. This is all within the S gene. And so the S gene variants from the B17, we actually spike it into the original Wuhan HU01 to get the full genome for this particular Accuplex construct. And this table shows the different amino acids, the nucleotide changes, and 3 different constructs that we have in our first product. Again, this is the Accuplex full process reference material. Construct 1 has all of these X's of the mutations that are included. Construct 2 is the South African, and construct 3 is the Brazil, Japan. You can see the X's mean all those mutations are included. So now you have a choice of using, detecting any of these constructs, these mutations across the S gene with a different tube. And I've just highlighted in yellow here 2 of the mutations I've talked about throughout the presentation, E484K, which you can see is in these 2 constructs, and the D614G, which is actually in all 3 of these constructs. D614G seems to have higher infectivity in some in vitro assays. So this is the SARS-CoV-2 Accuplex constructs that we have. Now we actually have 2 product formats. The first product format is the Accuplex, which I just talked about, and we'll have the positive reference material. So we have these 3 constructs plus the Wuhan, the original Wuhan genome. It's 1.5 mils, about 5,000 copies per mil in the universal transport medium. And we also include a negative reference material, which is the human RNase P. This is RUO labeled. And for sequencing, we've developed the SARS-CoV-2 RNA mix. What's different here is that in the single tool, we have 80 variants across the whole genome. So there's 38 mutations in the S gene, 16 mutations in ORF1A and ORF1B, 26 in 3A, E, M, etc., as you can see. So again, this is now designed for NGS applications. And particularly important will be determining the limits of detection for these assays. So we're actually making RNA mixes at these allelic fractions, 1%, 5%, and 20%. So you can start testing the LODs for your assays. Concentration, again, 5 million copies per mil. You can see the nanograms per mil here in tris, in a background of GM243A5 cell line extracted RNA. In QC released qubit digital to determine copies per mil, and then we use NGS to confirm the variants. So both of these products are just in R&D right now. Please contact us at LGC SeraCare for more information about these products. So now I'd like to welcome Nate back to the webinar, and we can do a Q&A. And again, thank you very much for your attendance, and I look forward to some questions.
And I want to thank you, Dr. Garlick and Dr. Ledeboer, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in from our audience members. Now, are there any current mutations or strains in particular that you are concerned about?
I will let Russell answer that one. Has he connected back into us?
And I think we're just waiting for him to reconnect. How about if I go ahead and ask another question? Are the Ocuplex ref materials or purified RNA for genotyping or sequencing to be used differently, or are they interchangeable?
That's going to be another Russell question, Susie. So I think we'll wait until he comes back on that one as well.
Okay. Let's see here. Let me move on to another question, and I want to thank our audience for their awesome questions coming in already. Who says that SARS-CoV-2 can be isolated from blood? As you mentioned, the various specimen limitations for quantitating respiratory viruses, is there any possibility of seeing blood-based quantitative tests in the future?
So this is a great question, and there's been a number of investigations into using not only blood, but a number of other specimen types to improve the diagnosis for SARS-CoV-2. We've seen data on blood, we've seen data on stools. Quite interestingly, the data on stools is very, very good, showing reasonably high sensitivity there. Blood ultimately depends upon the assay. We're seeing variable levels of sensitivity and variable levels of shedding of the SARS-CoV-2 virus within the serum. So I think we would certainly need some additional data and some additional study really following the kinetics of viral circulation in the blood before we move forward entirely with that, because I do think we would want to see some additional data around serum plasma, the duration of how long those patients would be viremic, the level of viremia, and truly whether that's associated better or worse with progression of disease in individual patients. So we've seen data that shows that it can be detected in serum. We really need to see some of that additional data of how well does that correlate with clinical disease, as well as how does it perform across multiple different assay platforms. What a great question.
Thank you so much, Dr. Ledeboer. And we'll continue to wait for Dr. Garlick reconnecting. Now, you cite the article that recommends checking LODs when a new variant becomes prevalent in local populations. In practice, what does that mean, Dr. Ledeboer?
So generally it can mean a couple of different things. So the first thing that you can generally do is, and if you're using it, it will differ if you're building your own test versus if you're working together with a commercial manufacturer of your test. If you're building your own test, you really need to be on a fairly regular basis taking your primer probes, searching them against all of the known sequences, and really making sure that those primer probes will amplify across the new variant sequences as well. It's also an important reason why when we develop tests for the SARS-CoV-2 virus, we don't target just a single gene. So we see many diagnostic tests that will have targets against the E genes, targets against the spike protein or the spike protein gene, targets against the nucleocapsid gene. And by using a multi-targeted approach, that really improves the chances of us finding it and decreases the chances of a single SNP or a single variant ultimately having a major impact on resulting or providing accurate results. Now, if you're working together with a commercial partner for your assay, the easiest way to do this really is in partnership with your commercial partner. They are required to, on a routine basis, perform surveillance, make sure that their assays are detecting variants and notifying customers. So the easiest way to do it with a commercial vendor is really working together with that vendor and partnering with them to make sure that they're doing that in a timely manner and that they're giving you documentation to be able to prove that information.
Thank you so much. And Dr. Garlick, welcome. I'll give you this next question, Dr. Garlick. Are there any current mutations or strains in particular that you are concerned about?
Yeah, great question. So again, from my local Massachusetts data that's being generated, the P1 has already been introduced into the population, so it's more infectious and perhaps has more disease outcome. So that's being closely watched, as well as the B1351 that originated in South Africa. So even within Massachusetts, there's lots of travel going back and forth. Those two are variants of concern that are being followed very closely.
Thank you so much. And Dr. Garlick, are the Accuplex ref materials or purified RNA for genotyping or sequencing to be used differently, or are they interchangeable?
Yeah, so the way I like to look at this is the purified RNA is very good in the initial stages of assay development and validation. And you can use it to standardize, optimize the reverse transcriptase step, and as well as the PCR step. Very good for that, and you can prove, you can detect these variants. It's a good positive control for that aspects. But as Nate mentioned during his presentation, we're dealing with respiratory viruses, and it's the whole workflow that you have to really think about the performance and precision reproducibility. In the Accuplex material, since it is a full process control, it's encapsulated virus, it's in universal transport meter, it's actually a better reference material to really monitor the assay over time and to prove for your laboratory, as you report out results, that your assay is performing appropriately. So they have different utility, but they're both very valuable tools for the laboratories.
Thank you so much. And again, I want to thank our audience for their participation. These are great questions coming in. Gentlemen, there are calls for investing in high throughput testing, example, greater than 50,000 to 100,000 tests per day at the regional or state level to address the issues of test capacity that we've encountered a year ago. What is the role of medical center testing and local public health labs supporting high throughput testing?
So Susie, this is a great question. I think this really goes to what can we learn from the result, or what can we learn as a result of this pandemic, and how do we make sure that we are adequately prepared for the next pandemic? Yeah. Because certainly one of the biggest issues that we've experienced early on, and thankfully this has somewhat subsided since then, is test availability and test capacity. And what we are largely going to see is we're going to need to see partnerships that are developed that include local medical centers, the national reference laboratories, as well as local and regional public health laboratories really partnering together to build this level of capacity within our healthcare system. one of the key things that we really learned early on was we relied on too few laboratories to ultimately do much of the early testing. Quite interestingly, if I look back a year from now, we were, we had been testing for about a month, but when we started to build our assay and we really started to look at how are we going to address this pandemic, we were told by the FDA, no, you can't perform testing because you haven't submitted anything yet in terms of your clinical trial, and you can't submit any data on performance of your assay until you've had a case. And that was incredibly frustrating, not only for our lab, but for many others. And so I think moving forward, we need to remove some of those regulatory barriers and also find ways that we can push that testing down into the clinical labs, into the reference labs, and support it through public health to make sure that we can address this moving forward. So a lot of it is going to be taking the instrument systems that we've already invested in and using those. It's going to be taking and utilizing those newer technologies like sequencing, which allow us very, very high degrees of throughput at very low costs, and being able to use those types of technologies and expand the use of those technologies. And it's also about getting those point-of-care diagnostics into the hands of our first-line healthcare providers as quickly as possible as well. So it's really got to be a multi-tiered approach that we have to take. And if you don't mind, I actually wanted to ask Russell a question based on one of his answers, if you don't mind.
Sure, of course.
So Russell, one of the things that's been kind of interesting that we've seen in one of my neighbors to the east in the state of Michigan is we've seen a massive expansion of COVID-19 in Michigan. And Michigan has been very, very hard hit throughout the pandemic. Now this really seems to be the B117 variant. Any thoughts as to why B117 is having such a dramatic effect in Michigan, but we're not seeing it in Indiana? We're not seeing it in Illinois, or even really in Wisconsin. It's been kind of an interesting phenomenon. Do we think that that is more of a localized patient, or more of a localized population issue, or do you think that there's something special about that variant?
It's a great question, great observation, Nate. When I think about the answer, I just look to the data in England, because it was identified early on in southeast England. But when they finally looked at the data across the regions, they realized it was moving pretty rapidly across all of England over the period of like 3 or 4 weeks. So I think you're correct. Yeah, Michigan is a hot spot right now. But I think next week or the week after, we're going to find it's in Illinois, it's in Indiana, it's in Wisconsin. It's already here in Massachusetts. It is more infectious. I've seen different numbers of infectivity, 1.5 or so more than the Wuhan strain. So we really have to, keep a close eye on the recommendations by the CDC for masking and all of the good things we've been doing to keep this pandemic under control.
Thank you so much, gentlemen. Now, Dr. Ledeboer, you brought up regulations and some of the barriers. Someone just did a follow-up question and said, how do you plan, or maybe all labs plan, to deal with those kinds of regulatory barriers?
So I think, number one, we've already seen a shift the FDA really, they've gotten a lot of criticism, but they've actually responded very, very nicely. They're very, very quickly reviewing assays, getting them onto the market. They have really moved to the idea of trying to get things sped through the approval process as well. I think one of the smartest things the FDA did very early on was to say, okay, if you're using a modification of the CDC assay, you can fall under the CDC's EUA approval and you can go with it. It's substantially equivalent. But I think at the end of the day, how do we prepare this for the next pandemic? I think it's really going to come down to our professional societies, whether it be the College of American Pathologists, whether it be whether it be ASCP or the American Society for Clinical Pathology, AACC, AMP, AASM, any IDSA, any of our societies or all of our societies really coming to the FDA and saying, this is the framework that we need to build to respond to the next pandemic. And that's going to have to be a very collaborative effort. And it's going to have to be done also in collaboration with the WHO as well. So I think it's going to be, a multi-year conversation in terms of how do we change that regulatory framework so that it can be responsive. We've certainly also seen some interesting information coming out of the Inspector General's office of the FDA, really questioning whether the FDA has the ability to regulate lab-developed tests. As we continue that conversation a bit further, it will be interesting to hear, where we ultimately land on laboratory-developed tests, and depending on where that answer ultimately comes, that may be a very viable option for labs too going forward.
Thank you so much. And this next question, I think, is on the minds of everyone. How quickly is this virus mutating?
I think it's always mutating. And the question is are there any mutations that will end up being of high consequence? And so it remains to be determined. Just a little more background on this virus is that many of these mutations have come from immunocompromised patients, unfortunately, because it seems to reproduce effectively in immunocompromised patients who have extended periods of infectivity. So again, the more we can do to keep the virus out of the general population, especially the immunocompromised populations, the quicker we'll be able to get it under control. Nate, do you have anything to add to that?
Yeah, I would just say, if we look at some of the data on it, we're seeing about 7 new variants emerge. The data I recently saw was about 7 new variants a week. That certainly isn't the frequency of mutation. That's variants that ultimately survive. Because we have to remember, the vast majority of these variants that emerge ultimately are not favorable variants to the virus, and that lineage ultimately dies off. So it's certainly not as bad as it could be, but it's certainly, we are seeing a reasonable degree of mutation that's occurring.
Thank you so much. We have time for one more question, gentlemen, and we'll kind of continue our discussion about regulations. You clearly show the importance of LODs. What changes or recommendations would you make to the FDA or the EUAs or other regulatory bodies to ensure assay performance meets the requirements for LODs and patient care?
So there's 2 really important things that are happening in this particular case, Susie. First of all, we've seen the first couple of 510K approved tests. BioFire, for example, went through and got 510K on their respiratory panel via the de novo 510K submission route. So we're beginning to see the FDA establish a standard for limit of detection for these tests. And I think, these early adopters or the early movers to go through the 510K process are really the pioneers, and we're going to see a lot of additional assays move through that traditional FDA review route. So I think that will be an important gating step. We've also seen, again, as we've seen more contemporary assays move through the approval process, there is a bit more standardization in terms of what would be accepted as the reference standard, how sensitive that test needs to be. And so we see ultimately a bit of a more rigorous comparison that's occurring. Again, the key issue with this, though, is balancing the speed at which we get assays onto the market, really responding to that issue of how do we make sure that we develop and have adequate capacity that's present on the market, with ensuring that we have good, accurate, and reliable assays that are on market. So I think it will be really important and really key for manufacturers moving forward to make sure that their assays are designed to be sensitive, that they will identify the different variants that emerge, and that the assays are, well suited for use in the field, if you will.
Dr. Garlick, Dr. Ledeboer, I want to thank you, gentlemen, for your time today and for your important research and your presentation. And before we go, I also want to thank our audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, LGC SeraCare, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years till April of 2023. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with colleagues who may have missed today's live event. Until next time, Stay safe and stay healthy, everyone. Take care. Bye-bye.